I once worked with a KOL who had strong reservations about a new drug we were promoting. I patiently listened to their concerns, provided data from clinical trials, and arranged for a meeting with our clinical team to address specific points. Eventually, the KOL became more open to the product based on the scientific data and discussions we provided. How do you balance the scientific and commercial aspects of your role as an MSL? Answer. Balancing two involves focusing on providing scientifically accurate and unbiased information while aligning with the company's goals. I ensure that my interactions with healthcare professionals are grounded in solid data while also understanding the commercial strategy to ensure that the right message is communicated at the right time. Conclusion. These questions span entry-level to advanced roles and cover a variety of areas, including. Certainly. Here are more advanced and role-specific interview questions and answers for different positions. 6. Clinical Research Coordinator. Easy questions. 28. What is the role of a clinical research coordinator? Answer. A clinical research coordinator, C, manages day-to-day operations in clinical trials. This includes recruiting participants, maintaining trial records, ensuring compliance with protocol, and collaborating with researchers to ensure data is accurate and reliable. What are the key responsibilities of a CRC in a clinical trial? Key responsibilities include participant screening, informed consent process, organizing and scheduling study visits, data collection and management, ensuring compliance with regulations, e.g. GCP, and communicating with study sponsors. Medium questions. 30. How do you ensure that all clinical trial protocols are followed during the study? Answer. I ensure protocols are followed by thoroughly reviewing the trial design and protocol with the study team, conducting regular training, and performing routine checks to confirm adherence to GCP, good clinical practice, and regulatory standards. 31. What measures do you take to ensure patient safety during a clinical trial? Patient safety is a top priority. I ensure all protocols are followed, monitor for adverse events, maintain regular communication with the investigator, and ensure the informed consent process is properly conducted. I also have contingency plans in place for any unexpected issues. Hard questions. 32. Describe a time when you had to deal with a non-compliant patient or site during a clinical trial. How did you resolve it? Answer. I once worked on a trial where a patient was not adhering to the medication schedule. I scheduled a meeting to discuss their concerns and provided additional education about the trial's importance. I also worked with the investigator to modify the follow-up schedule, ensuring the patient remained compliant without compromising safety. 33. What steps would you take if you noticed discrepancies in the data collected during a clinical trial? Answer. I would first identify the source of the discrepancies by cross-checking the data with source documents. I would document the issue and communicate with the study team for their investigation. If necessary, corrective actions would be taken, and additional training would be provided to site staff to prevent recurrence. 7 pharmaceutical regulatory affairs interview questions. Easy questions. 34. What does regulatory affairs in the pharmaceutical industry mean? Answer. Regulatory affairs involves ensuring that pharmaceutical products meet the regulatory requirements of the countries they are sold in. This includes navigating the approval process, ensuring compliance with local and international laws, and managing product registrations. 35. Can you explain the role of the FDA in drug approval? Answer. The FDA, Food and Drug Administration, reviews and approves new drugs for safety and efficacy before they can be marketed. They also monitor ongoing clinical trials and assess manufacturing practices, labeling, and post-market safety. 36. What is the importance of understanding the regulatory pathway when launching a new pharmaceutical product? Understanding the regulatory pathway is crucial to ensure that the product meets the requirements of regulatory agencies like the FDA, EMA, and other global authorities. This helps avoid delays, ensures patient safety, and allows for smooth market entry. 37. How would you handle a situation where a regulatory agency requests additional data for a new drug application? Answer. I would promptly gather the required additional data from relevant studies or trials. If needed, I would consult with the regulatory team to discuss the issue and develop a strategy for presenting the new data to the agency in a clear and timely manner. 38. Can you provide an example of a time when you had to deal with a regulatory agency's rejection of a submission? What was your response? Answer. In one case, a submission for a new drug was rejected due to concerns about safety data. I immediately conducted a thorough review of the data, identified gaps, and worked with the clinical team to generate additional studies. We resubmitted the data with additional evidence, leading to eventual approval. 39. How do you stay updated with changing global regulatory standards? Answer. I stay updated by subscribing to industry journals, attending regulatory affairs seminars, and networking with industry professionals. I also participate in webinars and engage in training programs offered by regulatory bodies like the FDA and EMA. 8. Pharmaceutical manufacturing specialist interview questions. 40. What is the difference between a sterile and non-sterile pharmaceutical product? Answer. A sterile pharmaceutical product is one that is free from microorganisms and their spores. Non-sterile products may contain microorganisms, but they are not harmful due to preservatives or the manufacturing process. 41. What is the role of a pharmaceutical manufacturing specialist? Answer. A pharmaceutical manufacturing specialist oversees the production process, ensuring that all operations comply with GMP standards. They ensure that equipment is properly maintained, manufacturing processes are efficient, and products meet regulatory requirements. Medium questions. 42. What steps do you take to ensure the quality of pharmaceuticals during production? Answer. I ensure quality by conducting regular inspections, implementing strict quality control procedures, ensuring that all equipment is calibrated, and ensuring compliance with SOPs, standard operating procedures. I also work closely with quality assurance teams to verify product standards. 43. What challenges do you face in pharmaceutical manufacturing, and how do you overcome them? Answer. Common challenges include equipment failure, raw material issues, and ensuring compliance with regulations. I address these by conducting thorough root cause analysis, implementing corrective actions, and maintaining close communication with other departments to ensure smooth operations. Hard questions. 44. Describe a time when you had to troubleshoot a production issue that was impacting product quality. How did you resolve it? Answer. In a production run, we discovered that one batch of tablets was underweight due to a malfunction in the granulation equipment. I worked with the maintenance team to identify the issue, recalibrated the machine, and ensured quality control measures were followed to prevent future discrepancies. 45. How would you approach improving the efficiency of a manufacturing line that is consistently underperforming? Answer. I would conduct a thorough assessment of the line, identify bottlenecks, and analyze each step of the production process. I would implement process optimization techniques like lean manufacturing, improve worker training, and ensure proper maintenance of equipment to increase overall efficiency. 9. Medical Science Liaison, MSL. Interview questions. Easy questions. 46. What is the role of a medical science liaison? MSL. Answer. An MSL is a healthcare professional who serves as a bridge between pharmaceutical companies and the medical community. They provide scientific support, educate healthcare professionals on the company's products, and gather insights to inform product development. 47. How do you maintain relationships with healthcare professionals? Answer. I maintain relationships by providing accurate, scientifically backed information, listening to their feedback, and maintaining consistent, professional communication. I also attend medical conferences and networking events to stay connected with the healthcare community. 48. How do you keep up with the latest research and developments in your field? I subscribe to medical journals, attend conferences, and network with key opinion leaders, KOLs. I also participate in company-led scientific webinars and review updates from regulatory agencies. 49. What is your strategy for handling challenging or skeptical healthcare professionals regarding new drug therapies? Answer. I engage by presenting well-researched, evidence-based information and answering any concerns they might have. If needed, I offer clinical trial data, safety profiles, and real-world evidence to support the therapy's benefits. Hard questions. 50. Describe a challenging interaction with a key opinion leader, KOL. How did you manage the situation? Answer. I once worked with a KOL who had strong reservations about a new drug we were promoting. I patiently listened to their concerns, provided data from clinical trials, and arranged for a meeting with our clinical team to address specific points. Eventually, the KOL became more open to the product based on the scientific data and discussions we provided. 51. How do you balance the scientific and commercial aspects of your role as an MSL? Answer. Balancing the two involves focusing on providing scientifically accurate and unbiased information while aligning with the company's goals. I ensure that my interactions with healthcare professionals are grounded in solid data while also understanding the commercial strategy to ensure that the right message is communicated at the right time. Conclusion. These questions span entry-level to advanced roles and cover a variety of areas including 427. What are the key responsibilities of a quality control QC department in a pharmaceutical company? Answer. The key responsibilities of the QC department include testing and ensuring the quality of raw materials, in-process materials, and finished products. This includes conducting physical, chemical, and microbiological tests to ensure that products meet the required specifications. The department also ensures compliance with GMP regulations and manages the release of products for sale by ensuring they are safe and effective. 428. What is analytical testing in pharmaceutical quality control? Answer. Analytical testing refers to the use of scientific methods to test the chemical, physical, and microbiological properties of pharmaceutical products. This includes tests for potency, purity, stability, and contaminants. Analytical testing helps verify that the products meet the specifications outlined in the manufacturing and regulatory standards, ensuring the product's safety and efficacy. Medium questions. 429. Explain the difference between stability testing and validation testing. Answer. Stability testing is conducted to assess how the quality of a pharmaceutical product changes over time under various environmental conditions, such as temperature, humidity, and light. The goal is to determine the product's shelf life and establish proper storage conditions. Validation testing, on the other hand, is done to ensure that a manufacturing process, equipment, or system consistently produces the desired output and complies with specified requirements. Validation is performed at the beginning of a new process or after significant changes to ensure it meets the necessary standards. 430. How do you handle the investigation of out-of-specification OOS results in QC? Answer. When faced with OOS results, I would initiate an investigation to determine the root cause of failure. The process begins with reviewing the testing procedures, equipment calibration, and dental factors. If the failure is reproducible, additional testing may be conducted, and corrective actions are taken, such as process adjustments or revising testing methods. Documentation of all investigations, findings, and corrective actions is crucial to maintain regulatory compliance. Hard questions. 431. Describe how you would ensure compliance with Good Laboratory Practices, GLP, in a QC lab. Ensuring compliance with Good Laboratory Practices, GLP, involves adhering to strict protocols regarding documentation, equipment calibration, sample handling, and testing procedures. I would ensure that all lab personnel are trained in GLP standards. All instruments are regularly calibrated, and all experiments are conducted according to validated methods. Furthermore, all data would be accurately recorded with traceable results, and any deviations from standard procedures would be documented and addressed promptly. Explain how you would set up a corrective and preventive action, CAPE, system in QC? Answer: setting up a CAPA system involves the identification of issues, such as deviations, nonconformances, or customer complaints, and implementing corrective actions to address the root causes. I would first assess the issue by conducting a thorough investigation, implementing short-term corrective actions, and then identifying long-term preventive measures. I would ensure that CAPA findings are documented and any changes made are communicated to relevant teams. Regular audits of the CAPA system would help track its effectiveness and ensure compliance. 76. Pharmaceutical data analysis interview questions. Easy questions. 433. What is the importance of data analysis in pharmaceutical development? Answer: data analysis is crucial in pharmaceutical development to assess the safety, efficacy, and performance of a drug throughout the clinical trial process. By analyzing large datasets, researchers can identify trends, correlations, and patterns that can guide decision-making. This ensures that drugs are developed in a way that maximizes their benefit to patients while minimizing risks. 434. How would you describe the role of statistical analysis in clinical trials? Answer: statistical analysis in clinical trials plays a critical role in determining whether a drug is effective and safe for use. It is used to analyze trial data, calculate the significance of results, and estimate the confidence intervals of treatment effects. Statistical methods, such as hypothesis testing, regression analysis, and survival analysis, help to interpret complex data, ensuring that clinical trial results are scientifically valid and reliable. Medium questions. 435. How do you ensure the integrity of data in pharmaceutical research? Answer: ensuring data integrity in pharmaceutical research involves maintaining accurate, consistent, and complete data throughout the research process. This includes using validated software systems, establishing proper data entry protocols, conducting regular audits of data, and training staff on good data management practices. Data should be recorded in real time, any changes should be tracked, and backup systems should be in place to prevent data loss or corruption. 436. What is the difference between descriptive statistics and inferential statistics in pharmaceutical research? Answer: descriptive statistics involves summarizing and organizing data to describe the basic features of a dataset, such as mean, median, standard deviation, and frequency distribution. It is used to give a clear picture of the data at hand. Inferential statistics, on the other hand, uses sample data to make generalizations or predictions about a population. It involves hypothesis testing, confidence intervals, and regression analysis, helping researchers make inferences about the broader population from a smaller sample. Hard questions. 437. How do you perform a regression analysis in clinical data analysis? Answer: regression analysis in clinical data analysis is used to model the relationship between a dependent variable and one or more independent variables. I would begin by selecting the appropriate regression model: linear, logistic, or Cox regression, depending on the data. The model would be fit to the data, and key metrics such as R-squared, p-values, and confidence intervals would be used to assess the model's effectiveness. The results help in understanding the strength of the relationship between variables, making predictions, and identifying potential factors influencing the outcome of the clinical trial. 438. How would you handle missing data in clinical trials? Answer: missing data in clinical trials is a common issue that can affect the validity of the results. Depending on the amount and pattern of missing data, I would use different strategies to address the issue, such as imputation techniques, e.g., mean imputation, regression imputation, or complete case analysis. If the missing data is substantial, I would consult with the clinical team to decide if a modified analysis plan is necessary. It is important to document and justify the handling of missing data to ensure transparency and compliance with regulatory standards. 77. Pharmaceutical regulatory compliance interview questions. Easy questions. 439. What is the role of regulatory compliance in the pharmaceutical industry? Regulatory compliance in the pharmaceutical industry ensures that drugs, devices, and biologics are developed, manufactured, and marketed in accordance with regulatory requirements set by agencies such as the FDA, EMA, and WHO. Compliance ensures that pharmaceutical products meet safety, efficacy, and quality standards to protect public health. It also involves maintaining proper documentation, conducting audits, and ensuring that manufacturing practices comply with industry regulations. 440. What are the main regulatory agencies that oversee pharmaceutical companies? Answer. The main regulatory agencies that oversee pharmaceutical companies include the FDA, Food and Drug Administration, in the United States, the EMA, European Medicines Agency, the MHRA, Medicines and Healthcare Products Regulatory Agency, in the UK, and WHO, World Health Organization, globally. These agencies regulate the development, testing, approval, and post-market surveillance of pharmaceutical products to ensure their safety, efficacy, and quality. Medium questions. 441. Explain the concept of good manufacturing practices, GMP, and its importance. Answer. Good manufacturing practices, GMP, are a set of guidelines and regulations established to ensure that pharmaceutical products are consistently produced and controlled to meet quality standards. GMP covers all aspects of production, including raw material sourcing, equipment, facility cleanliness, and documentation. The importance of GMP lies in ensuring that products are safe, effective, and of high quality, reducing the risk of contamination, defects, and non-compliance with regulatory standards. 442. What is Drug Master File DMF, and why is it necessary? Answer. A Drug Master File is a confidential document submitted to regulatory authorities that contains detailed information about the manufacturing processes, raw materials, and control mechanisms for a pharmaceutical product. It allows drug manufacturers to share proprietary information with regulatory bodies while keeping sensitive information confidential. DMFs are necessary to demonstrate that the manufacturer's processes comply with GMP and regulatory standards. 443. How do you ensure compliance with international regulations when launching a drug globally? Answer. To ensure compliance with international regulations, I would thoroughly research the specific requirements of each country or region where the drug will be marketed. This involves understanding the regulatory guidelines for clinical trials, product labeling, packaging, and... Sure, here are additional theory questions and answers related to biotechnology, vaccines, and various associated fields, expanding further on the topic. 31. What is the difference between attenuated and inactivated vaccines? Answer. Attenuated vaccines contain live pathogens that have been weakened so that they cannot cause disease in healthy individuals. These vaccines typically generate a strong and long-lasting immune response. Example, the BCG vaccine for tuberculosis. Inactivated vaccines contain pathogens that have been killed or inactivated through heat or chemicals. While they are safe and stable, the immune response is usually weaker than that of attenuated vaccines, and multiple doses may be required. Example, the inactivated polio vaccine. 32. What are monoclonal antibodies, and how are they used in biotechnology? Answer. Monoclonal antibodies are laboratory-made molecules that can mimic the immune system's ability to fight off harmful pathogens, such as viruses and bacteria. They are made by creating identical copies, clones, of a unique white blood cell. These antibodies are widely used in treatment, diagnostics, and vaccine development. They can target specific cells or proteins associated with disease. For example, monoclonal antibodies like rituximab are used to treat certain cancers and autoimmune diseases. 33. Explain the process of gene therapy in the context of biotechnology. Answer. Gene therapy is a technique used to treat or prevent diseases by introducing, removing, or altering genetic material within a person's cells. The goal is to correct faulty genes responsible for disease development. Gene therapy can be used to treat genetic disorders such as cystic fibrosis or hemophilia by delivering functional genes into a patient's cells. There are two main types: somatic gene therapy, targeting body cells, and germline gene therapy, which affects reproductive cells, though it is still highly controversial. 34. What are CRISPR-Cas9 gene editing technologies and how do they work? Answer. CRISPR-Cas9 is a revolutionary gene editing technology that allows scientists to make precise changes to DNA within living organisms. CRISPR, clustered regularly interspaced short palindromic repeats, works by using a guide RNA to direct the Cas9 protein to a specific location in the DNA. Once at the target site, Cas9 cuts the DNA, allowing researchers to add, delete, or alter genetic material. CRISPR has huge potential for treating genetic diseases, enhancing crops, and exploring gene functions in various organisms. 35. What is bioremediation, and how is it used in environmental biotechnology? Answer. Bioremediation is the use of living organisms such as bacteria, fungi, or plants to remove, neutralize, or degrade contaminants from the environment. This process is commonly used to clean up polluted environments, including oil spills, heavy metals, and other toxic chemicals. By leveraging the natural ability of certain organisms to break down pollutants, bioremediation provides a sustainable and environmentally friendly alternative to chemical cleanup methods. 36. What are biosensors and how do they work? Answer. Biosensors are analytical devices that use biological components to detect and quantify substances such as pathogens, toxins, or pollutants. They work by incorporating a biological material, e.g., enzymes, antibodies, or cells that interacts with the target substance. This interaction is then converted into a measurable signal, which can be read by the sensor. Biosensors are widely used in fields like medicine, e.g., glucose monitors, environmental monitoring, and food safety. 37. Explain protein expression in biotechnology. Answer. Protein expression refers to the process by which cells produce proteins based on the genetic instructions encoded in their DNA. In biotechnology, researchers often use recombinant DNA technology to insert a gene of interest into a host organism, e.g. bacteria, yeast, or mammalian cells, to produce large quantities of a specific protein. This process is used to produce therapeutic proteins, enzymes, and vaccines. Example, insulin produced using E. coli bacteria. Thashner, 38. What is biotechnology fermentation, and why is it important? Ants fermentation is a biological process in which microorganisms like bacteria, yeast, or fungi convert organic compounds like sugars into chemicals, gases, or alcohol. In biotechnology, fermentation is essential for the production of a wide range of products, including antibiotics, vaccines, and biofuels. The controlled fermentation process enables large-scale production of desired substances while maintaining quality and consistency. 39. What are biopharmaceuticals, and how are they different from traditional pharmaceuticals? Answer. Biopharmaceuticals are medical drugs produced using biotechnology, typically derived from living organisms. These include monoclonal antibodies, hormones, enzymes, and vaccines. They are different from traditional small molecule pharmaceuticals, which are chemically synthesized. Biopharmaceuticals often have more complex structures, require special storage and handling, and are used for treating chronic diseases, cancers, autoimmune diseases, and other conditions. Number 40. What are stem cells, and what is their role in biotechnology? Answer. Stem cells are undifferentiated cells that have the ability to develop into various specialized cell types. There are two main types of stem cells: stem cells, which can differentiate into any cell type, and adult stem cells, which are more limited in the types of cells they can become. Stem cells have significant potential in regenerative medicine, tissue engineering, and drug development, as they can be used to replace damaged or diseased tissues and organs. 41. What is pharmacogenomics, and how does it relate to personalized medicine? Answer. Pharmacogenomics is the study of how an individual's genetic makeup affects their response to drugs. It plays a key role in personalized medicine by enabling the development of tailored drug therapies based on genetic profiles. This ensures that patients receive the most effective medication with minimal side effects, as certain genetic variations can influence how a drug is metabolized or how well it works. 42. What is the significance of vaccine adjuvants in immunization? Answer. Vaccine adjuvants are substances added to vaccines to enhance the body's immune response to the antigen. They help increase the strength and duration of immunity, ensuring better protection with fewer doses. Some common adjuvants include aluminum salts, oil in water emulsions, and lipid nanoparticles. They are especially important for vaccines targeting diseases that require a strong immune response, like influenza and hepatitis B. 43. What are gene drives and how are they used in biotechnology? Answer. Chain drives are genetic systems that increase the likelihood of a gene being passed on to the next generation, overriding typical Mendelian inheritance. They are used to spread beneficial traits rapidly through populations, such as in controlling mosquito populations to fight diseases like malaria. Gene drives have the potential to revolutionize pest control and genetic conservation, but also raise ethical and ecological concerns. What is the role of biomaterials in medical devices and regenerative medicine? Answer. Biomaterials are materials designed to interact with biological systems for medical purposes. They are used in medical devices, such as implants, prosthetics, and scaffolds for tissue engineering. Biomaterials can be natural, e.g. collagen, or synthetic, e.g. polymers, and must be biocompatible, non-toxic, and able to integrate with living tissue. In regenerative medicine, biomaterials provide a scaffold for new tissue growth or stimulate healing in damaged organs. There are 45. What is synthetic biology and how does it relate to biotechnology? Answer. Synthetic biology is an interdisciplinary field that combines biology, engineering, and computer science to design and construct new biological parts, devices, and systems that do not exist in nature. It involves the modification of organisms and cellular pathways to create new functionalities, such as bacteria engineered to produce biofuels or drugs. Synthetic biology has applications in health, agriculture, and energy production. These are more advanced theoretical questions that span across various topics in biotechnology and related fields. Each question provides an opportunity to dive deeper into the specific areas of research, application, and innovation in biotechnology, vaccines, and other life sciences. Let me know if you'd like to explore more questions or any other area in detail. Here are more advanced and challenging interview questions, along with detailed answers, focusing on biotechnology, biopharma, data analysis, and related fields. Hashna 126. Explain the concept of monoclonal antibodies, MABs, their production and applications in therapeutics. Monoclonal antibodies, MABs, are antibodies that are made by identical immune cells, all derived from a single parent cell. They are engineered to target a specific antigen or epitope, making them highly specific and effective for targeted therapies. Production. The production of MAbs begins with the immunization of a mouse or other animal with the target antigen. The animal's immune system produces a variety of antibodies against the antigen, which are then harvested. The next step involves creating hybridomas, a fusion of the animal's immune cells with a myeloma cell line, creating immortalized cells that continuously produce the desired antibody. The antibodies are then purified and formulated for therapeutic use. Applications in therapeutics. Cancer treatment. MAbs can be used to target cancer cells specifically, either by blocking growth signals, marking the cells for immune attack, or delivering toxic drugs directly to the cancer cells. Autoimmune diseases. MEMAbs are used to inhibit overactive immune responses in diseases like rheumatoid arthritis and Crohn's disease. Infectious diseases. MEMAbs can be used to neutralize viruses or bacteria, providing targeted treatment. For example, palivizumab is a MAB used to treat respiratory syncytial virus, RSV, infections in infants. Cardiovascular diseases. Some MAbs are used to prevent heart attacks or stroke by targeting specific proteins like PCSK9, which regulates cholesterol. 127. What is the difference between biosimilars and biologics, and what challenges do biosimilars face in the market? Answer. Biologics. These are large, complex proteins or other substances derived from living organisms used in the treatment of diseases. They include monoclonal antibodies, vaccines, recombinant proteins, and cell therapies. Biologics are typically more expensive due to the complexity of their manufacturing processes. Biosimilars. Biosimilars are highly similar versions of an already approved biologic, the reference product. Although they are not identical, biosimilars have no clinically meaningful differences in terms of safety, purity, or potency from the reference biologic. Challenges faced by biosimilars. Regulatory hurdles. The approval process for biosimilars is rigorous, requiring evidence of comparability in terms of quality, efficacy, and safety to the reference product. The process involves clinical trials and a demonstration that there are no significant differences in immunogenicity. Market acceptance. Although biosimilars offer cost savings, doctors and patients may be reluctant to switch from a well-established reference biologic due to concerns about efficacy or safety. Patent issues. Biologics are often protected by patents, and biosimilar manufacturers must navigate the intellectual property landscape to avoid patent infringement. Manufacturing complexity. Since biologics are produced in living cells, even small changes in the manufacturing process can lead to differences in the final product. Ensuring consistent quality in biosimilars is therefore a major challenge. Reimbursement. Some healthcare systems may have less favorable reimbursement policies for biosimilars, making it harder for these products to penetrate the market. What is the significance of next-generation sequencing, NGS, in personalized medicine? Answer. Next-generation sequencing, NGS, refers to high-throughput sequencing technologies that allow for the rapid sequencing of entire genomes, exomes, or transcriptomes. NGS enables the identification of genetic variations and mutations at a much faster pace than traditional methods like Sanger sequencing. Role in personalized medicine. Genomic profiling. NGS allows for detailed genomic profiling of patients, identifying genetic mutations, single nucleotide polymorphisms, SNPs, and copy number variations that may contribute to disease. Targeted therapies. By understanding the genetic mutations in a patient's tumor or disease-causing genes, NGS can guide the selection of targeted therapies. For example, mutations in the EGFR gene in non-small cell lung cancer can be treated with EGFR inhibitors. Pharmacogenomics. NGS can help determine how patients will respond to drugs based on their genetic makeup, aiding in the selection of the most effective drugs with the least side effects. Cancer. NGS allows for the identification of driver mutations in cancer, enabling precision oncology, where therapies are tailored to the specific genetic profile of a patient's tumor. Rare genetic diseases. In patients with undiagnosed rare genetic disorders, NGS can help identify mutations that traditional diagnostic methods may have missed, providing a path toward diagnosis and treatment. 129. How do you approach the design and optimization of experiments for evaluating drug efficacy and safety in preclinical studies? Answer. Designing and optimizing preclinical experiments to evaluate drug efficacy and safety involves a systematic approach. 1. Define objectives. First, clearly define the research objectives. This could include determining the efficacy, whether the drug works, safety, whether the drug is safe, and mechanism of action, how the drug works. 2. Choose appropriate animal models. Select animal models that closely mimic human disease. For example, for cancer drugs, xenograft models or transgenic mice may be used to evaluate tumor response. 3. Dose selection. Establish the dose range for testing based on pharmacokinetic data. The dose should span a therapeutic range along with a margin to observe potential toxicity. Toxicity studies will determine the maximum tolerated dose, MTD. 4. Endpoints and biomarkers. Define primary and secondary endpoints, e.g. tumor shrinkage, survival rate, and identify biomarkers, e.g. blood levels of the drug, biomarkers of toxicity. 5. Control groups. Use both positive control, known treatment, and negative control, placebo groups, to assess the relative efficacy and safety of the new drug. 6. Statistical power. Ensure that the study is statistically powered to detect meaningful differences. Use appropriate statistical tests to analyze the data and reduce false positives negatives. 7. Safety monitoring. Continuously monitor the animals for adverse effects, e.g., weight loss, behavioral changes, organ damage throughout the study. Histopathology and clinical pathology studies should be done to assess internal damage. 8. Pharmacokinetics and metabolism. Measure the drug's blood concentration, half-life, and how it's metabolized or excreted in animal models. 9. Replicability. Perform studies with sufficient replicates to ensure the results are reproducible. 130. What are the most important ethical considerations when conducting clinical trials, particularly in vulnerable populations, e.g., children, elderly, patients with severe illnesses? Answer. Ethical considerations in clinical trials are critical, especially when dealing with vulnerable populations. 1. Informed consent. Ensure that all participants or their legal guardians understand the purpose, risks, and benefits of the trial. For vulnerable populations, this involves making the consent process simple, clear, and ensuring they are fully aware of their rights. For minors or those with impaired decision-making capacity, consent must be obtained from a legal guardian or proxy. 2. Minimizing risk. Trials should be designed to minimize harm and ensure that the risk is justified by the potential benefit. Vulnerable populations are more susceptible to adverse effects, so special precautions should be in place. 3. Monitoring. Vulnerable populations require additional monitoring to ensure safety. Regular health assessments and early adverse event detection are critical. 4. Equity in selection. Ensure that vulnerable groups are not exploited. Participants should be chosen based on scientific necessity and not convenience. For example, a trial should not only include elderly people simply because they are easier to recruit. 5. Confidentiality. Protect the privacy of the participants by ensuring that data is anonymized and securely stored. 6. Equitable treatment. Ensure that vulnerable participants are not subjected to undue coercion or undue pressure to participate. 7. Post-trial access. After the trial ends, ensure that participants have access to any effective treatments or ongoing care, especially if the drug proves to be beneficial. These are additional hard questions that were deep understanding and critical thinking about advanced. Here are additional advanced questions with answers related to pharmaceutical quality control and assurance. 156. What are the challenges involved in implementing good manufacturing practices, GMP, in a pharmaceutical company? Answer. Good manufacturing practices, GMP, are critical to ensuring that pharmaceutical products are produced consistently and meet quality standards. However, implementing GMP can be challenging for several reasons. Challenges. 1. Compliance with regulatory standards. GMP requirements vary across different regions, e.g. FDA in the US, EMA in Europe, making it difficult for multinational companies to maintain consistent compliance across all markets. 2. Training and personnel management. Continuous training of employees is necessary to maintain compliance with GMP. Staff must be regularly updated on new guidelines and changes in regulations. 3. Maintaining documentation. GMP requires thorough documentation at every stage of production. Maintaining proper records, auditing documents, and ensuring traceability can be cumbersome and time-consuming. 4. Process control and validation. Adhering to process control and validation procedures is essential to ensuring product quality. Any deviation from validated processes needs to be properly investigated, which can increase operational complexity. 5. Equipment maintenance and calibration. Regular maintenance and calibration of equipment are required to ensure the equipment is functioning as intended. This can be resource-intensive and requires significant planning. 6. Quality assurance and audits. Continuous internal and external audits are necessary to ensure GMP compliance. Companies may face challenges in preparing for and passing audits, particularly when it comes to addressing nonconformities. 7. Supply chain management. Ensuring that suppliers also adhere to GMP standards can be difficult. A company must manage raw material quality, storage conditions, and transportation methods, all of which impact GMP adherence. How to overcome these challenges: regular audits and internal inspections, continuous employee training on GMP standards, employing automated systems for documentation and record keeping, establishing a strong supplier management program, ensuring proper equipment calibration and maintenance schedules. 57. What is the significance of process analytical technology, PAT, in pharmaceutical manufacturing? Answer. Process analytical technology refers to a system for designing, analyzing, and controlling pharmaceutical manufacturing processes through timely measurements of critical quality attributes, CQAs. That enables real-time monitoring of the process to ensure that the product meets its specifications. Significant 1. Real-time monitoring. P enables real-time data collection on critical parameters like temperature, pressure, humidity, and concentration, allowing for immediate adjustments to ensure the product meets quality standards. 2. Enhanced process understanding. It helps pharmaceutical manufacturers gain a deeper understanding of the manufacturing process, thereby improving process control and reducing variability. 3. Improved product quality. By continuously monitoring and adjusting the process, P ensures that the final product consistently meets quality specifications, improving the overall quality and reducing the likelihood of defects or rework. 4. Increased efficiency and cost savings. PAT enables continuous monitoring and optimization of processes, which can reduce waste, lower production costs, and shorten development timelines. It also minimizes the risk of batch failure, leading to fewer recalls or product issues. 5. Regulatory benefits. Regulatory bodies like the FDA and EMA encourage the use of PAT in manufacturing to enhance the product's quality and consistency. Companies implementing PAT can gain regulatory flexibility and faster approvals. 6. Continuous manufacturing. PAT is essential for continuous manufacturing systems, as it ensures that the process is tightly controlled and quality is maintained throughout production. Examples of PAT tools: spectroscopic techniques, e.g. near-infrared spectroscopy, NIR, chemometric modeling, inline process analyzers. Key benefits: real-time data for quality control, enhanced process flexibility, higher product yield, and fewer process interruptions. 158. How do you define process control, and why is it crucial in pharmaceutical manufacturing? Answer. Process control refers to the monitoring and control of the variables involved in the manufacturing process to ensure the consistent production of high-quality products. It involves the use of data, sensors, and automated systems to track critical process parameters and make adjustments to maintain the desired product quality. Importance of process control. 1. Ensures product quality. Effective process control ensures that the product consistently meets the required specifications for identity, strength, quality, and purity. 2. Minimizes variability. It helps reduce variations in the manufacturing process that can lead to defects or deviations from quality standards. 3. Improves efficiency. Process control enables manufacturers to optimize their production schedules, reduce downtime, and make the best use of resources, leading to cost savings and higher throughput. 4. Compliance with regulatory standards. Regulatory bodies such as the FDA and EMA require that manufacturers implement robust process control systems to meet GMP standards. This ensures consistent quality and safety. 5. Enhances traceability and accountability. Process control helps maintain a clear record of each step in production, making it easier to track and trace issues if deviations or product failures occur. 6. Cost efficiency. By optimizing the manufacturing process and reducing defects, manufacturers can lower waste, minimize rework, and reduce material costs. Components of process control. Automated data collection. Sensors and automated systems collect data on critical process parameters such as temperature, pressure, and humidity. Feedback loops. Systems that automatically adjust process parameters based on real-time data. Predictive modeling. using historical data to predict future outcomes and adjust processes accordingly. Quality control checkpoints, routine inspections and tests at various stages of the process to ensure that the product remains within specification. 159. Describe the role of lean manufacturing principles in the pharmaceutical industry. Answer. Lean manufacturing is a set of principles aimed at minimizing waste and maximizing value in the manufacturing process. Lean focuses on reducing inefficiencies, improving processes, and delivering high-quality products at lower costs. In the pharmaceutical industry, lean principles can significantly enhance productivity, reduce waste, and improve product quality. Role of lean manufacturing in pharmaceuticals. 1. Waste reduction. Lean principles help identify and eliminate sources of waste, e.g., overproduction, excess inventory, transportation, waiting times that do not add value to the product. 2. Continuous improvement. Lean focuses on continuous process improvement, Kaizen, fostering a culture of efficiency and optimization in every aspect of production. 3. Just-in-time inventory. By using JIT principles, pharmaceutical companies can reduce excess inventory, which can reduce storage costs, minimize expiration risks, and improve cash flow. 4. Increased flexibility. Lean allows manufacturers to be more flexible and responsive to changes in demand, regulatory requirements, or supply chain disruptions. 5. Improved quality. Lean principles emphasize error reduction and continuous quality improvement through regular process evaluations and employee involvement in quality assurance. 6. Employee engagement. Lean encourages team-based problem solving and gives employees a sense of ownership over the manufacturing process. This leads to improved job satisfaction and productivity. Examples of lean techniques. 5S, sort, set in order, shine, standardize, sustain. Organizes the workspace to improve efficiency and reduce waste. Value stream mapping analyzes the flow of materials and information to identify inefficiencies. Kanban, a visual scheduling system that helps manage inventory and production flow. Total productive maintenance, TPM, focuses on maintaining equipment and preventing downtime to improve process reliability. Benefits in pharmaceuticals: reduced costs, faster time to market, enhanced product consistency, improved regulatory compliance. Explain the importance of analytical method validation in the pharmaceutical industry. Answer. Analytical method validation is a critical process that ensures the reliability, accuracy, and reproducibility of analytical testing methods used to assess the quality of pharmaceutical products. This process verifies that the methods used for testing meet the required criteria for their intended purpose. Importance of analytical method validation. 1. Ensures accuracy and precision. Validation confirms that the analytical method provides consistent and accurate results, ensuring that the pharmaceutical product meets its specifications. 2. Regulatory compliance. Regulatory agencies such as the FDA, EMA, and ICH require that analytical methods used in drug testing are validated to ensure they are reliable and capable of detecting impurities, active ingredients, or contaminants. 3. Reliability of test results. Validated methods provide reliable and reproducible results, reducing the risk of false positives or negatives in testing. 4. Improved quality control. Analytical method validation is key to ensuring that pharmaceutical products are of the highest quality and meet all safety and efficacy standards. 5. Product development. During drug development, validated methods are used to assess the purity, potency, and stability of active ingredients and finished products, helping researchers optimize formulations. Key parameters of method validation. Specificity. The method must measure the target analyte without interference from other substances. Linearity. The method must produce results that are directly proportional to the concentration of the analyte over a specified range. Ensures that only products that meet quality standards are released to the market. Compliance with regulatory requirements. Proper handling of OOS results helps maintain compliance with GMP and regulatory standards. Risk mitigation. Addressing root causes reduces the risk of recurrence and prevents product recalls, improving customer trust. Weiner Hertog 197. What is the role of process analytical technology, P, in pharmaceutical quality control? Okay. Answer. Process analytical technology is a system used to monitor, control, and optimize manufacturing processes in real time. It involves the use of analytical tools to gather data and inform decision-making, enhancing process control and ensuring consistent product quality. Key elements of PAT. 1. Real-time monitoring. PAT uses sensors and data analysis to monitor critical process